Hello and welcome to SAM Biology class. Friends, today we shall discuss about horizontal gene transfer. The transfer of genes from parents to progeny is called vertical gene transfer. Vertical gene transfer in eukaryotes also introduce genetic variation into population. With each new generation, recombinant progeny are formed due to gametic fusion during sexual reproduction. Prokaryotes, however, do not reproduce sexually. So it is quite common to think that in population of prokaryotes, the genetic variation is relatively low. But this is not the case. Prokaryotes have evolved three different mechanisms for creating recombinants. These different mechanisms are collectively known as horizontal gene transfer or HGT. Horizontal gene transfer is also known as lateral gene transfer, which is distinctive from vertical gene transfer. In HGT, genes are transferred from one independent mature organism to another, often creating stable recombinants that has characteristic of both the donor and the recipient. Horizontal gene transfer has been important in the evolution of many species. One example of HGT is that prokaryotes sharing an ecological niche, for example the human gut, exchange genes, and this alter the nature of the microbial community in a habitat. Second example is the transfer of group of genes encoding virulence factor from pathogenic bacteria to non-pathogenic ones. Finally, horizontal gene transfer has played an important role in the evolution and spread of antibiotic resistance among pathogenic bacteria. In horizontal gene transfer, a piece of DNA called exogenode is transferred to a recipient cell. DNA transfer can occur by three different processes. They are number 1, conjugation, number 2, transformation, and number 3, transduction. In conjugation, a close physical contact between the donor and the recipient cell is essential. DNA transfer takes place through a specialized secretion system. In transformation, a competent bacterium takes up DNA fragment from its environment. Finally, transduction is the bacteriophage-mediated DNA transfer from one bacterium to another. If the exogenode has the genes that are already present in the recipient, then the recipient cell will be temporarily diploid for these genes. Such a partial diploid cell is called as merozygote. The exogenode has 4 possible fates. In one case, the exogenode has sequence homology with the recipient cell's chromosome. It is because of the sequence homology the exogenode is stably integrated into the recipient cell's chromosome. This gives rise to a stable recombinant which reproduce and form a population of stable recombinant. In second case, when the exogenode is self-replicating, for example, if it is a plasmid, then it persists outside the endogenode, but because it is self-replicating, so as the recipient cell reproduces, it gives rise to a population of stable recombinant. In third case, when the transferred DNA is not able to self-replicate, Neither it is sequence homology with the recipient cell's chromosome. In this case, as the recipient reproduces, the exogenous is eventually lost from the population, so stable recombinants are not formed. Finally, in some cases, there is a process called as host restriction, because of which nucleases present in the recipient cell degrade the exogenous into pieces, thereby preventing the production of stable recombinants. So friends, this is about horizontal gene transfer. For more updates, please subscribe my YouTube channel SAM Biology Class, and also please share your comments. Thank you very much.